In this video, we are going to discuss an important topic, that is chemical and biological material handling. In every laboratory environment, handling chemicals and biological samples safely is extremely important. Improper handling can lead to accidents, contamination, health hazards, and even damage to the environment. So, in this session, we will understand the correct handling procedures, safety practices, storage methods, and waste disposal systems followed in laboratories. Let's begin. By the end of the session, all participants should be able to understand safe handling practices for both chemicals and biological materials. We will also learn about different types of laboratory hazards and how to identify them. Apart from that, we will discuss proper storage methods, labeling requirements, biosafety measures, chemical safety, and waste disposal procedures. Finally, we will understand the importance of GLP compliance in laboratory operations. In laboratories, we regularly work with different types of materials such as chemicals, biological samples, reagents, microbial cultures, and other hazardous substances. Because of this, proper handling becomes extremely important. Safe handling helps in protecting laboratory personnel from injuries and exposure. It also ensures accurate experimental results, protects the environment, and helps laboratories comply with regulations and safety standards. Laboratory chemicals are classified into different categories based on their properties and hazards. Corrosive chemicals can damage skin and surfaces. Flammable substances can easily catch fire. Toxic chemicals may affect human health if inhaled or consumed. Reactive chemicals may react and with substances. Oxidizing agents can increase the intensity of fire. Each type of chemical requires specific storage and handling procedures for safety. Along with chemicals, laboratories also handle different biological materials. These include microorganisms, cell cultures, blood samples, tissue samples, and clinical specimens. Such materials may contain infectious agents or contamination risk. Therefore, proper biosafety practices must always be followed while handling biological samples. Before handling any chemical, it is very important to identify its hazards. Laboratories use labels, hazard symbols, material safety data sheets, and safety classifications for this purpose. These systems provide information about the risks associated with chemicals. Understanding these hazards helps prevent accidents, exposure, and unsafe handling practices. Material Safety Data Sheets, also called MSDS, are very important safety documents available in laboratories. They provide complete information about chemical properties, associated hazards, safe handling procedures, storage conditions, first aid measures, and spill management. Before using any unfamiliar chemical, laboratory personnel should always check the MSDS. These documents must be easily accessible inside the laboratory. To ensure chemical safety, certain standard handling practices must always be followed. Personnel should wear proper PPE like gloves, lab coats, and goggles. Chemicals should be used only in properly ventilated areas. Direct contact must be avoided. All chemicals should be handled carefully and according to SOPs or standard operating procedures. Following these practices protects both laboratory staff and experimental samples. Biological materials should always be handled using aseptic techniques to avoid contamination. Biosafety cabinets are commonly used while working with infectious samples. Sterile equipment and proper disinfection procedures are also essential. Controlled laboratory practices help minimize contamination risk and reduce the chance of infection. Personal protective equipment, commonly called PPE, acts as the first line of protection in laboratories. Essential PPE includes lab coats, gloves, face masks, safety goggles, and closed footwear. The type of PPE used may vary depending on the material being handled. Proper use of PPE helps prevent exposure to hazardous chemicals and biological agents. Chemical storage should always be done carefully and systematically. Chemicals must be stored according to compatibility, hazardous classification, temperature requirements, and ventilation needs. Proper labeling is very important. Incompatible chemicals should be stored separately to avoid dangerous reactions. Secure storage cabinets must also be used. Incorrect storage can lead to serious laboratory accidents. Biological materials require controlled storage conditions. Samples should be stored at appropriate temperatures and in sterile environments. Proper labeling systems should be maintained for easy identification and traceability. Access to storage areas should be restricted to authorized personnel only. Proper storage helps maintain sample integrity and laboratory safety. In case of any chemical spill or exposure incident, immediate action is necessary. Laboratory personnel should be informed immediately. Spill control kits must be used according to emergency SOPs. Appropriate PPE should be worn while cleaning spills. Contaminated materials should be disposed of safely. Quick and correct response helps reduce laboratory hazards and prevents further accidents. Laboratory waste must be disposed of carefully to protect people and the environment. Chemical waste should be segregated according to hazard type and disposed in an approved containers with proper labeling. Biological waste should be collected in biohazardous bags and usefully steriled by autoclaving before disposal. Improper waste disposal can create serious health and environmental problems. Laboratories are classified into different biosafety levels based on the risk associated with microorganisms. BSL 1 is used for low risk organisms. BSL 2 is for moderate risk agents. BSL 3 laboratories handle airborne pathogens. BSL 4 laboratories work with highly dangerous organisms requiring maximum contaminant. Higher biosafety levels require stricter safety and contamination measures. Good laboratory practices or GLP plays a major role in safe laboratory operations. GLP requires proper labeling systems, controlled storage conditions, SOP-based handling procedures, regular personal training, and complete documentation. Maintaining proper records and traceability requires quality, consistency, and regularity compliance in laboratories. Proper material handling provides many important benefits. It improves laboratory safety and reduces contamination risk. It also improves data reliability and increases operational efficiency. Most importantly, it helps laboratories maintain regularity compliance with protecting personnel and the environment. To summarize today's session, we discussed the safe handling of chemical and biological materials. We understood hazard identification methods, PPE usage, biosafety practices, storage systems, and waste management procedures. We also discussed the importance of GLP requirements in laboratory operations. Proper handling of materials ensure safety, quality, and compliance in laboratories. Thank you everyone for your attention and participation. I hope this session helped you understand the importance of safe chemical and biological material handling. If you have any questions or doubts, please feel free to ask them in the comment section. Thank you once again.